So I'm Andrea Matlock, and I'm part of Jenny Van Eyck's group at Cedars-Sinai, and we've been doing the proteomics for Neurolinks, and for which we've created data both on a minimal amount of data at the infancy or the initiation of the Neurolinks center. And so we did iPSCs, but the larger component of the Neurolinks program was the iPSCs that were differentiated into motor neurons. And so that's the bulk of the data set that we've provided. And we were talking about getting this workshop going and having an open discussion and everything. So welcome everyone. Thanks for being here, and we'll go on to the next.
I can go next, I guess. My name is Mike McCoss. I'm part of the Broad Proteomics project, although in the last 6 months or so, I haven't been as actively involved with it. I've been more focused with the Proteomics Working Group and finalizing some of the overview papers, the SciData papers, although that's gone slower than I'd like as well. The overview manuscript is completed, I believe, and then the letter to the editor has been drafted. The plan is to submit them, just so everybody knows, this weekend, and then so anytime after that, the other individual papers can be submitted as well. We'll see how this goes. We haven't had communication with the editor for a little bit since Jake was involved, but we'll still just go forward, and if not, we can reach out to someone like John Yates, maybe at JPR, I think, who would maybe receptive as well. So we'll still go with the initial plan at SciData and see how that goes, because I think it is kind of ideally suited for that mechanism.
Thanks, Mike. I can go next. I'm Jenny Van Eyck, and Andrea and I work together on the Neurolinks. And if Vinit's on the call, Vinit's also part of us, but I don't see him yet.
Yeah, we don't have many people yet. I don't know what's going on.
I think people are having a hard time finding the breakout rooms,
yes.
But it's okay.
Okay, well, hi everyone. I am Malvina. I am part of Broad Proteomics. Our focus here for the past 6 years were the two targeted assays, P100 and GCP. And over the past 3 years, I guess now, with Sebastian, we have been working on DIA analysis methods. He's leading it, I'm following.
Yeah, just to complete that, yeah, I'm working also here in Malvina in the same office. And our focus has been on method development of DIA and then developing tools for automating the DIA analysis. So before we used to do everything, curating the data by hand, and then we had the challenge of creating tools to automate all of that. And then Karen tomorrow is going to present something where we are developing a pipeline in the cloud to automate the analysis of DIA. And yeah, that's what we've been working on.
Hi, this is Chris Swanson-Fisher from NINDS. I'm the project officer for Neurolinks, taking over from Dr. Mark Sutherland since she departed to CZI. And I've worked with Jenny primarily through AMP-PD efforts. Good to see you, Jenny.
Hi there. Is anyone else on the line? Do not be shy. Only us. Okay.
Well, it does look like there are other people, but I'm not sure. Yes, but if maybe people aren't stepped out or not. So Andrew, do you want to maybe remind us what the agenda was that had been kind of worked out, or?
Yes, absolutely. So what we had discussed, and I think the bottom line is we want to make this a open and fun discussion. I mean, this is the conclusion of the Links program, and certainly it would be fun to talk about ways that we might work together and further showcase the links data and how that might work. And so a component of this can be left open to discussion, but I think what's been highlighted today in the various presentations from Melvina's group and others is that the implications and sometimes unexpected cases in which the links data has been used for different research projects, in particular Dane Mayfield on the alcohol addiction from the L1000 data. So we wanted to kind of formulate this in that to start some discussions at least is to talk about the large data and the importance and values of our links centers and the data that we've provided, and talk about how or each center come up with a phrase or elevator pitch that you would do to a senator or congressperson to highlight the benefit of your center's data. And a second component would be another challenge for us to talk about how we might consider mining each other's data sets in various ways and come up with new biology spins on that. And I think if we did something like that, it might be fun to get that published in a form where that's a perspective or something or other, depending on the outcome of that. That would help showcase what we've done and all the work that is available and letting the broader community know that they have access to this data.
So Malvina and Sebastian, just kind of put a little context to this, is when, you know, when Jake and I were first talking about doing the P100 data, we kind of made a decision to try to collect the data by data independent acquisition, even though we knew it was going to be essentially a targeted assay. But the goal there was always potentially reanalysis of the data. And I know that's obviously what you guys have been thinking about. But one of the values of it is having maybe other people reanalyze existing data sets. And so one thought we were chatting the other day is one great demonstration, at least in the proteomics field, was how can we use each other's data sets maybe to answer different questions than they were originally intended. That's always been the promise of the data collection that we're using, but no one's ever done it. No one's ever taken somebody else's data set and found something new in it and then wrote a paper on it. Even though everybody talks about that's what the value of it is. So I don't know. I thought that could be a just, you know, point. One thing I also wanted, this is again very specific to just the sort of the P100, is there plans from the Broad to continue data production after links on P100?
Not really. Yeah. I think because most of the people have left by now. And we have finished with all these drug perturbations. So the last year, actually, we focused more on the neuroside with Lewy size group to be able to address biological questions rather than just do more tranches of drugs. And we added this parasite cell line in the pool of samples. We also screened some neurons, microglia, but these were without drugs. So we have, like, from H9 cells, the last data set we recorded was different cell lines, differentiated cell lines, NPCs, parasites, astrocytes, microglia, neurons, without any drug perturbations. And we are drafting a couple of papers up with them. One is at the more advanced stage, the Alzheimer's story. And then we'll see. But no, I don't think, Mike, that there will be any more data production.
So we are, as you know, we are
yes
have to do the P100 in our side. We are using a slightly different automated sample prep protocol than you are. And because you guys sent us those cell lines, we are reaching the extra complexity of how do we calibrate between the assays. But that's at some point in time, it would be great to actually have a discussion with you and Sebastian and other people there on what we're seeing and the best way to calibrate between it. But that's another discussion. But I thought I'd bring that up because one of the things we wanted to discuss today is how do we extend the lifespan of the data that we've collected here. And one, of course, the values of it is the library that you've already built with all these perturbations. Then it becomes of value to generate new data to potentially be able to map back to that existing set of signature library. So at least we will have it operating here. We hope to be, and we hope to be able to help disseminate that to other people as well. But you guys help disseminate it to us, so I guess we can kind of pass it forward. I guess. Yeah. So yeah. So I think I think those are kind of things that we would like to. There are other people that have joined. Do people want to maybe introduce yourselves? We're kind of discussing predominantly between some of the Neuralinks and the Broad proteomics efforts, but it'd be great to kind of get people to at least introduce yourselves who are joined.
I'll introduce myself. Nicholas Clark from University of Cincinnati.
Okay.
I'm a PhD student here.
Great. Nice to meet you. Great.
So maybe I will introduce myself.
Okay.
I'm Berta Marowska. I currently postdoc in Albert Einstein College of Medicine.
Okay.
But I was working with, I'm working as a data analyst and I'm working with proteomics data. And I was using the very first, I think, version of CMAP for one of my publications. So.
Okay, great.
This is Ajay Pillai.
Yeah, Ajay, how are you?
All right, how are you?
Great. So the other thing I think, Malvina, that's gotten me at least interested that you mentioned, I don't want to, is the pipeline aspect that you're working on. We're actually obviously doing similar sorts of things as well. I'm wondering at some point in time, as a separate thing, we should maybe just touch base on what the different efforts are. We are building everything around Cromwell now, so that I think should have some influence of being able to align things with what the Broad's doing. And we can maybe pass back kind of some notes, and the same thing too with Jenny's group as well. Obviously there are I think implications, I think they're broad based between all the proteomics with the data that's been collected both from Neuralinks and also from the P100 side, that again, if we were to analyze data with different tools and if we can harmonize some of those pipelines, those workflows a little bit better, or make them at least transferable, that would help, I think. That's always been the goal here that we've wanted, and it's never been that easy. We've spent a lot of time doing this, and so I know everybody has. Yeah.
What should be the first step to do that, to try to make that happen?
Yeah. I think we should find people, and I think it would be of enormous value to have somebody from another group work to analyze another group's data. I don't know. And we can do that in the context of, it could even just be reanalyzing some of the P100 DIA data. I don't know. I'm just trying to throw it out. I don't want to, say somebody else will be reanalyzing the data that Malvina has been producing, but I think that's kind of part of the goals, right? Is to make it reanalyzable.
Yeah, and I think being transparent and having meetings around it, and being able to highlight some things, right? If that's somewhat something that we're all interested in committing some time to and working together on, it would be interesting.
Yeah.
And further validation, right, of everyone's analysis, I think.
Yeah.
So absolutely, we are
So,
in.
yeah. So I think we should pick a starting point of that. Maybe even just a plate. It could be a plate that from the Broad. And they've done tons of plates, but maybe have different groups to analyze a plate of samples, and it could even be that way. It doesn't overlap, Malvina, with some of the stories that you're trying to put together right now with some of the papers. It could be a plate that's already
Just hanging around?
Yeah.
It's actually all the plates with drugs. I don't. We can analyze any of the plates with the drugs, and Sebastian has already. You will see in Karen's presentation tomorrow. We have analyzed a few plates. How many are those, Sebastian? 9, 12?
Yeah, we have a data set with 9 plates that each one has 96 replicates. So we could choose one, like we have been focusing on one that's MCF7 samples with the kinase inhibitor tranche of drugs that we've seen is of good quality. So I would say that could be a starting point.
Does anybody else have another data set? I don't want to volunteer, obviously, the entirely the Broads data set, which is great. There's nothing wrong with the Broads. Obviously, it's great, fantastic data. I'm just trying to say, like, if there's somebody else has another data set that they would like to see. So, and for those who have just joined in, one of the things that we've thought that it could be an important kind of closing thing that has been obviously the promise of proteomics data and the data generated by links is be able to have other people, consumers of the data, make use of it beyond the links project. And one thing that's always been a promise of collecting the data by data independent acquisition or SWAT, right, which is how Neuralinks and at least the Broad collected the data, is that it could be reanalyzed to find new things. And so and everyone says that that's the promise of it. In fact, you read the intro of almost every single one of these papers, they say that the data can be reanalyzed or a grant proposal, but it's very rarely done. And so I think it would be a great contribution on showing that it can be done and that new things were found. And with the P100 is particularly valuable because the data has only really been released using basically 96 probes, right? When we know that there's thousands of phosphopeptides in the data set.
Yeah, I think it would be really super interesting to do. I think we could certainly check on one of the analysis as well. I was just trying to think about our DIA data, what would be of value. And I'm even thinking within the answer ALS. So, because it might be more interesting. We can release, of course, the DI data from Neuralinks. It's just not a lot of cell lines. And we have, well, I guess there's two things. One is we have a set of cell lines where they're all at different stages of differentiation, the same lines, but differentiated for multiple different times. I'm not sure that that's so interesting. Around the biology, there is a set of lines that we're having in the main manuscript, which is the Neuralinks ones, which is, can't even remember now if it's 4 lines of C9 versus, but we do have, the other thing we have, and I don't know, I'd have to get it cleared, but we have from the answer ALS, the first 67 samples. They were run on the 5600 and we're going to rerun them on the 6600 triple top to match the rest of the 1,000 samples that we're running for them. But that's an independent data set. And again, the triple top data is very different from when we run DIA on the thermo instruments, right? We run them DIA on both instruments and they are synergistic. They don't cover, of course, the same peptides. They do give different data, but we could provide that. It would be the Neuralinks iPSC derived motor neurons, an X number, but it's triple top data. Yeah. For sure. That's all we've run on this. And we did not analyze for PTMs or isoforms as it currently stands.
So one of the things that Ajay just sent around is an RFA specifically for pilot projects to enhance the usage of Common Fund generated data. So there is a specific request for applications and kind of doing this type of thing. So
This might be a little tricky. I don't know who's eligible for this. I haven't, at least in the first iteration, I don't think you guys were eligible for it. Maybe Andrea would be since she's no longer out of links.
Yeah.
I don't know.
Okay.
But just check that and let me know if I can assist with poking people in comment.
Thank you.
Absolutely. Yeah. Okay. Yeah, so I think, so I guess I haven't read it in detail, but I guess what I was just getting at is that it's supposed to be from groups, not from links initially, right? So, so anyway, we should look at it in more detail, but that RFA is definitely a possibility to look in.
Well, I'll just add too, I just think that, yeah, this would be a fun, as I said before, it would be a fun activity, a challenge for us to do internally among proteomics, the field, and just highlighting what we could do. And to extend upon what Jenny said, we're we're using TOF data from the SciEx instruments, triple TOF data. And so we've pushed things through knowing that there's a lot of information there that in a high throughput manner in which we're extracting certain things. And so just like the P100 data, we know that there's a much greater depth of information that is there. And so if we were to use, you know, just thinking about the different links data levels, if you think about the data levels, then we can talk about analyzing each one of these groups, maybe in using different programs and showing differences in that. And there's endless ways that we could start to approach this if we want to get organized and say, well, here's the possibilities, but what do we think is going to be the best way to kind of highlight the best of the data by multiple different search algorithms or data types.
So I can propose, I guess, then maybe two types of activities. So one is obviously a purely computational, where there's a plate that Sebastian and Malvinas and et cetera have analyzed or reanalyzed, that we can maybe have a couple other groups try to analyze, and then we can kind of come back together and do a compare and contrast. Maybe each person gives a 2 or 3 slide update on what the conclusions were of those data files. That's one. And then I know which is a little bit harder one for people to do is, but we at least would like to, we're going to actually redo, in fact, we've chosen to do MEK inhibitors as a first trial beyond the set of samples that Malvina sent us. And the reason why we chose MEK inhibitors is because there is some drugs that you guys have already done that are MEK inhibitors, but they're also they have different off-target effects.
Mm-hmm.
So the idea is, could we take different MEK inhibitors and get basically the same signature back? But we noticed that they won't be exactly the same, right? And that basically also shows that the value in the system's working.
Yes, so let me rephrase that. So what you're going to try to do to see in the complexity of the on and off target effects of each drug, whether or not you can find the common pathway by using multiple inhibitors, correct?
That's correct, yeah.
Nice.
So,
actually, it's really, I wish Karen had presented first, then we had this discussion, because we are going to touch on all these things tomorrow.
Okay, great.
I mean, yeah.
Yeah, so I think but I think the one thing that's a little different is that we will be using a different sample prep to try to map things back to the your existing P100 data, which is an extra complexity, I think. But yeah. Okay, so again, I think there's even more people who have joined. I'll mention, because I know Dusika is on the call. So one thing is, I think is the manuscript, I think is done. The letter to the editor has been drafted. The plan is to submit it by the end of the weekend if there's nobody else has any final comments on it. And then I will then let everybody else know to have follow-up papers. We're submitting everything to SciData still. And depending on you know, it's been a while since we've communicated with the editor. We might as well just at this point submit the papers. And if there is any issues, we will then reach out to someone like John Yates at Journal of Proteome Research or something like that to consider another submission. But I think SciData is still the most appropriate place for these papers.
Oh, that's great. Thank you, Mike, for doing that. And let me know if you need any help with anything to get that out.
I think right now, so the only thing I would like to do is, before I pull it out of Google Docs, is just I'll reformat and add the references to it. So that's the only thing I have left to do, I believe. So otherwise, I think you've left it in pretty great shape. So that was and it's had a bunch of people gone through it. So I know Malvina added a bunch of comments, and but I want to give people one last chance to look at it, because I'm not sure all of the groups have looked at it in as much detail as they should.
Yeah, I will definitely look through it as well.
Hey,
Thanks,
great.
Yeah, and thanks, Dusika, great job. I have looked through it, but I haven't added anything to it, or any comments. I was wondering too on how we might coordinate all of them together. Mike, do you have an idea of what we should expect of, should we just put them all on the Google Drive and
so what I've done in the past year, and Jenny maybe correct me for other of these sorts of linked papers is the first paper in should mention the other ones, which I will do in the cover letter, and then that's it. Then after that, then the other one, people can submit their links referencing saying it is one of those linked papers, right? So so that's the only thing that needs to be done. So at this weekend, I will submit the overview paper, and then at that point in time, everybody else should be free to submit their papers as well.
Yeah, you just need to link it. Mike needs to link all of our papers, and then we need to link when you submit just to his.
That's right.
Yes.
It's just in the cover letters. That's all.
Where are these cover letters, Mike? Do you have them centrally in the Google Drive?
I don't, but I will add that. Yes.
Okay, to find the editor's information and stuff.
Yep.
And yeah.
Yep.
Thank you.
Absolutely.
So are we all using the same cover letter, or that doesn't?
I think it should be, the cover letter should be specific to the individual papers
Right.
that are
Right.
being mentioned, but they should mention that it's part of the broader links proteomics working group discussion. So.
Will be good to get them in.
Exciting times, yes.
So are there other things beyond those kind of two topic areas that we could do? So Jenny, I do like the idea of the ALS IPS C cells too. I mean, that may be another way to reanalyze data.
Yeah, so we have the pipelines to do it across both, like I said, the thermo instruments and the triple TOF instruments, because depending on what we're doing, we flip between the two instruments. So, but they're not the pipelines aren't quite the same. So I'm not sure if other people want to try the triple TOF data. It is different. It's less accurate, but you get more peptides, kind of. So just, it has a very different feel for it. Wouldn't you say that, Andrea? You've run both.
Yes, so it would be interesting if anybody wanted to deal with the triple TOF. But yeah, it of course depends on the method used and everything, but we definitely get some overlap between them and some unique. So it's nice to see how each of them kind of highlight slightly different things.
So happy to share that data.
Does anyone want SciAx triple TOF data?
I think so.
So I think maybe we should just start some discussion about this and maybe have a follow-up that are a little bit more specific. I think there's clearly some reanalysis of data that could be valuable, and there's also and I think all of this demonstrates value to the links data production efforts, right? Which I think is helpful not only for us long term, but also helpful to the NIH program staff who obtain resources for the data production.
So I'll ask a question. So people doing DIA, how is your post-translation modifications going? We have like methylation, acetylation, succinylation, but darn if we can get phosphorylation to work well. I don't know. I'm not sure if anyone else is doing DIA PTMs.
I mean, I'm sure Sebastian has thought about this more recently than I have, but the site localization is definitely a major problem in all of the cases, right? I mean, I would have thought the same thing with methylation and acetylation too.
So we worked to get an algorithm, a scoring system for that. That was actually what took us. The paper went in, and then that of course was a question, and so we spent a lot of time actually, and we managed to get one. It was a lot of work. We had George working on, and also Brian. So we ended up, Brian got further along and actually did a really good job. So he managed in his system now to have it for methylation, acetylation, succinylation, a scoring system that based on known sites that we had synthesized, we're doing a good job on calling them correctly. But of course, I think they can be improved, but he did a good job. But yeah, the phosphorylation we haven't, we're just, we can't we can't get it right now. Although I know people are publishing that they have. But the scoring algorithm's a big part. We spent probably a good 8 months on that for our last paper. And it's like 5 lines in the paper.
So I know from when we had done some of the phosphorylation that one of the things that was pretty eye-opening was seeing that almost every phosphopeptide was a positional isomer of some form. And when you look at the data closely, that the only way when you did DDA, you triggered on the first one and then you didn't see it again. So you never sampled the different forms. But when you collected by DIA data, you did see all the different forms. But it's not always easy to localize the fragments to a specific form. So I don't know. Maybe Sebastian can come. I know he's been looking at this more.
Yeah. So
Thanks,
what
Sebastian.
we're using is a Spectre. That is the software developed by Ryan Beckner, who used to work in the lab and then left in the middle of the project. But his idea is to try to deconvolute the spectra by linear algebra. So the way he described it is to some sort of this algorithm is like Shazam. So it has this entire library of spectra and tries to match the intensities that are in the library to the data that you acquire in DIA. And what he did to demonstrate that his algorithm was working well for phosphopeptides was taking positional isomers, synthesized positional isomers, and then he could demonstrate that he can separate coeluting peptides because he's trying to deconvolute the spectra. So actually, the publication came out 2 years ago.
Yeah, we saw that, yeah.
So that's what we are using for P100 data.
So is there a localization score, Sebastian? Just curious, sort of like an A score type equivalent. I know that's what Earl came up with. He came up with something similar to that. He came up with and he did try to come up with a false discovery value for the localizations measurement. Because some you can easily localize, some you can't.
So no, there's no A score directly, but it's if we have in the library a spectra for both position isomers, then
it will compete them with one another.
It will match. So that's the problem, like having a high quality spectra for every possible isomer. So that's the problem that we're having.
Yeah. Good. I'm glad to know that we're not the only one. It's always good to know. Makes you feel better as you try to figure it out.
So, the only other thing that we were thinking about potentially discussing here today was I know Malvina, Jake, and you and a lot of other people in the group thought a lot very carefully about how do you assess the quality of your data sets. And I know even with the data that you guys sent us, there was a very detailed PowerPoint presentation slides on the. I know you probably don't remember it, but there was a bunch of there was some nice slides on how you assess the quality of the data before sending it out. And one thought we also discussed with between Jenny and Andrea is that's always an important thing about trying to make use of public data is having some measure of what the data quality is beforehand. And I'm not sure that that's made entirely clear, with all the links data of what's done to assess the quality of it. as I know of it because I've participated in your lab's calls and other things too, and I've seen measures of how you've kind of assessed it. I don't know if and we've thought very carefully about how do we extend that into the P100 that we're doing, and we've had, you know, beyond that, and then, but at the same time too, we also have things that our lab has done that's a little bit different than what you guys have done. I don't know if you, is there something we can do more broadly as kind of as a group of people who collected very heterogeneous different assay types, can use to report and think about what is the quality of someone's data? Things that are either done as far as system suitability, process controls. So there's also getting the language even straight between people. It's like, you know, we think of things as system suitability, process controls, and quality controls as like 3 separate things. And a lot of people just kind of have a single common term for things. And so that's something else I definitely would like to, you know, find a way to chat about here in this context. Is one is, how do we try to present a way that shows the quality of the data? That was something we're trying to get across in some of the SciData papers as well. I noticed everybody has some sort of metrics for what the quality of their data is, but everyone's using different metrics. So anyway, I'm kind of rambling now on just, but I think this is a good discussion topic in general for data that was collected under links. How do we?
I think it's super important because I think more and more people are starting to do larger data sets. So, you know, we're doing AMP-PD, as was mentioned, and we have the Gates Foundation now. So I think we have to, as a field, I think we should come up with something common, and maybe we can have, you know, increasing stringency. So the minimum, which means you have to have it, which should still be really, really rigorous, but then all the way up, because I think it's going to be important as these large data sets come out that people can understand and talk the same language, Mike, and have a context. I think if not, then it's going to be very hard, not just on proteomics, but on people trying to use the proteomics data, which is just a huge stumbling box, and people analyzing the data when they try to combine it with proteomics, right? So I think if we don't get it right, I think it's going to backfire on our field.
Yeah, I mean, I can at least share something separately that our lab does to kind of in some of this context. And this is, again, a little bit different. I'm not trying to say this in separately, but we tend to think about these things as there is a sample that's run every certain number of injections to assess the system, right? And this is a set of peptides which should be the same and confined, and there's a set of quantities that we have as a metric for precision and reproducibility, intensity, retention time, et cetera, et cetera. We then have things that are process controls, where we basically add a protein prior to digestion, and we have some peptides afterwards that we use to assess, is each run, is those peptides that are post-digestion, those are pre-digestion, did the digestion sample prep go okay? When in doing the P100, we're adding a protein that's a phosphoprotein prior hand to, so we actually demonstrate that the digestion worked okay and the enrichment of the phosphopeptide worked okay.
Can I just ask you a question? How did you buy that? Is it 100% phosphorylated? Did you phosphorylate it? Just curious how you did that.
We're actually just using casein, right? Casein, right?
Okay, that's pretty good.
Basically 100% phosphorylated. It's easy to use, but it tells you right off the bat if a digestion or one of the stuff is.
Yeah, no, it's nice.
In each plate, we actually have a common reference and a quality control. And these are 2 different samples that are pools. And this is very similar to what Malvina does in the P100 and the Broad does, where they basically have the same drug treatments like DMSO, and I think it's Storosporin, and there's also an HDAC inhibitor, I think, too, that's added to every single plate, right? And so the idea here is you have something that you can normalize everything to if you wanted to across plates, and then you have something that you should always have the same differences on each plate, right?
I have a question, Mike.
Yeah. Sorry, go ahead, Andrea. I'll say something after you.
I was just wondering if you've been analyzing casein for a long enough time that you've noticed any shift in phosphorylation from batches, and that you then just start working off of the next batch when you have to shift.
So we've done that for enolase. For unphosphorylated things, we add enolase, yeast enolase to each sample. And in there, we haven't seen much of a difference, but we also made a huge set of aliquots when we first done it. We haven't gone into a different one. Each day is a new thing for us picking out when someone does an experiment.
Yeah, we use beta gal for the same one. So we do the same. Ours is almost the same. Probably the proteins are different, but how we do it's the same. The only other thing that we added for links, and then we've certainly added for Answer ALS, is we have the batch controls. And Andrea, you can probably say this better, for actually the when they're in the iPSCs, we have a control for them, just so that we can monitor their performance at the iPSC when they drive the motor neurons.
Yeah, we do a batch, what we call a batch differentiation control and a batch technical control. So the batch technical control is a differentiated motor neuron culture that is made in bulk that we analyze each time. And then they use that same exact cell line to differentiate alongside of each sample set that gets differentiated, because with Answer ALS, we're trying to work up to 1,000 different patient lines. So we're working with different differentiated human genetic backgrounds for those cell lines. And so each time that same cell line is differentiated. So we're looking at it as the batch differentiation control and the batch technical control with each set we get.
Yeah, and the way I think about this common reference and the quality control is very similar to how the clinical labs use they use a calibration curve. They normally, if you're running vitamin D, right, you're going to run each batch, you're going to run a calibration curve, and you're going to have a low, medium, and high QC. And here we don't want to have a low, medium, and high QC for every single
Analyte.
Sample analyte, right? So we have one sample that's we know is a different sample. We have known differences in that sample that we've measured in every single batch, right? So you go through the quantitation, and when you're done with the quantitation, that batch has a set of known features that are different in a known level. So it flags the batch as being something went wrong with the quantitation if there's something.
Do you think there would ever be consensus that, so this is what we're doing for MPD, this is what we're doing for Gates, the same, this is identical. And if there would be any way to get journals to, just to accept kind of a technical design like that, so that just like you have to upload your data, you have to upload your QC data and show that you've met these criteria. Do you think there would ever be an agreement by journals to do that? Or do you think it's even?
It's always possible to write something like a perspective paper on this, right? On what's needed. But I find sometimes sometimes perspectives are viewed more than just having a scientific paper, right? So if you have a scientific paper, it's like, all right, it's another lab's how they do their controls, right? But if you have a perspective paper across multiple different labs, advocating for something like that, then it ends up becoming a little bit more strong. Sometimes with MCP, they will try to come up with these standards methods, and they'll have different committees where they set aside, these are what we believe should be some sort of standards. So I do think, those are also possible. I know when MCP started to get lots of data independent acquisition papers, they actually had a workshop specifically saying, we don't even know how to review them. We don't know how to assess them. People are all over the place as far as the tools. And so there was a discussion on having just some basic groundwork on what even need to be turned in. Nobody agreed on the final results, but everybody agreed through right?
That it needed to happen.
Yeah. So there's a starting point, right? That needs to be done. Anyway, so then I just want to point out too, this is what we are doing with the P100. We do actually have a DMSO and a treated reference. We're using Storosporin as well as one, and we add casein at the beginning. We then add heavy peptides and then medium peptides to assess the enrichment as well. So there is two separate process controls. We add PRTC is also added post enrichment to assess align chromatography and that we get the systems working okay. So that's all measured in each sample. Anyway, I just kind of wanted to point that out. I'm not trying to think of anything more specific than that, other than I think it is worthy of having a discussion here on when we do release data, we should be more upfront with why we believe the data is of certain quality. And I'm not saying we should force everybody to conform to a standard method, but I do think people should have methods for assessing the steps in which, and that everything worked okay.
I agree.
Because I've been in discussions with Jake's lab, for instance, where they've pointed out how plates have gone bad, right? And they've actually looked through the plates and they said, look, we need to redo this plate.
Or a pipette tip on the automation system doesn't pull what it's supposed to be pulling, and you have.
Actually, now that we are towards the end, we pulled all data together from DMSOs that we have in each plate. So we have 4 separate tranches of drugs with astrocytes, with NPCs, with pericytes, and we were really interested in seeing what the differences are. But there, what you end up doing is normalizing across all plates your data, right? And we got like beautiful clustering across plates also. But there is always a question mark every time you process a plate.
You know, I mean, that itself should be a paper. So we did this with Rudy Abersol's data. So we took the same exact perturbation that he did in yeast. He published a data set in yeast. And he actually even said that they can't compare their triple quad data versus their triple TOF data. We took their data and said, look, if you normalize it to their one common sample line that they did, they could compare between the different platforms. And then we took our data on an Orbitrap, normalized to our reference, even from different digestionists, different labs, different instrument platforms, and demonstrate that everything basically falls on a line. But here, I actually do have a slide here showing that. But I do think that would be, the more we can get people doing things like that, I think is absolutely fantastic. So here,
yeah, that was nice. I remember you talking about that last year.
Yeah. Yeah. Here, so we did. So this is their data. This is their 2015 data by SRM. This is their swap data for the same one. If we normalize it to the same reference sample, this is their own data, which they said they couldn't compare because they were different scales. And then we took our lab data and their lab data, and then when we normalized it to the common reference, it also fit on the line. These are different platforms, different people in different labs prepping it, but they prepped the same sample. The same person prepped the same reference standard using a different method, right? So any peptide that would be reduced by 75% would be reduced by 75% in the reference too, in theory. So anyway, I think these are underappreciated things, right? That I think that need to be made more clear, and the more examples of it, I think the better. Anyway, this is exactly why we use a common reference. There still will be batch effects, right, that occur, but it should minimize them. And I know there is an effort within the NCI to come up with a common NCI 7 sample that could be prepped by all groups all the time when they do a cancer sample set, which could potentially be used as a common calibrant between labs and groups and methods.
Yeah, so if Corey Bryston was on this call, he would say you have to be careful because that's a biological one. It's going to be hard to accession. So meaning you can't necessarily, it's not indefinite.
Yeah, but
right?
NIST, so to go against that, NIST has solved these problems, right? So every time they come up with a reference and that reference runs out, they then calibrate the old reference to the new one. And the example I always give people is like PD belemite, when we used to do carbon isotope ratios, right? Everyone normalizes to this fossil that no longer exists, but everybody has reference samples to what that value was.
So my caveat to that is the story of cardiac troponin I, of course, right? Where when they had to harmonize 20 different assays, they had a big competition, and people made, cloned it, did all kinds of stuff, made synthetic peptides to the epitopes. And the one that won actually was isolated from a human heart. And it was heavily, heavily modified. And it was the only one that would harmonize everybody's assay. So it turned out that it wasn't the pure protein actually was not good because biology, it's a heavily modified protein, and that's what's circulating in your blood when they're trying to measure it. So I think I would say the only caution to that is that you can still have PTMs and that can still mess you up.
Yeah, and I think Andy Lufo here has a great example too, it's just lipoproteins as well, right? If you actually have a plasma sample and normalize it to that, a specific one, that ends up being better. Anyway, I just want to comment I think that's a strength, and you should find a way to make sure that you report that, even just as a quick technical thing, saying, look, we have this ability to calibrate between plates.
Really nice.
It'd be nice, yes. Actually, I can show you something that, can I share my screen for a second?
Yeah, I think so.
Sorry, can I, oops, sorry, stop share, wrong screen.
That's always the problem with multiple screens, is that whenever you share it, it's like, which one
I'm
am I
sorry,
sharing?
which one? Do you see this one?
Yes, I see it.
You see this? Okay, awesome. Okay, let me put it in present. Because usually I have a yellow screen around it, but I don't see it. Okay. So actually these were data that we have acquired, NPCs, pericytes, neurons, and astrocytes throughout the years. And these are in triplicates. You see here DMSO treatments of NPCs in different plates. And this is a similarity matrix. So one question that came up during our meetings with Liwei Sai's group was, does even DMSO induce like changes to the cell lines? Because we don't really know, or is it the same? And we screened NPCs alone or NPCs with DMSOs, and these are from all different plates, and you see that they cluster really well together. This is a similarity matrix. But we were really wondering, until we saw that, we were like, okay, how the data are going to look across plates and across years? And then we did the pericytes. So you see actually big blocks of high similarity correlations here, and neurons and astrocytes. And you can even see here astrocytes with DMSO. We harvested in 2 different batches.
So
and you can definitely see the difference.
So this is your average for each cell line. What would be interesting to know is, if you look at your analytes that are high abundance, medium abundance, and low abundance, and see whether or not you get more of a spread with low abundance than you get with high abundance. I don't know if that would be true.
I see.
You know what I mean? But it would be interesting to know, because this is really stunning, right? This is your, yeah.
Yes, and
really
actually,
yeah.
This is a slide where I say that the S10 is included. Sometimes these are very high and they just drive the whole clustering. So we always try with S10 or without S10 marks in there. And actually, oh, this is GCP data now that I'm using it. Actually, this is GCP, I'm sorry, it's not phosphodata. I just recall this is why the S10 came up from histonase 3. But in general, these are things that we typically do on like for all of our data.
Yeah, I think it would be super interesting, especially the concentration and the effective matrix, right? I mean, you have the effective matrix here in the study, so that would be just really interesting to know.
Mm-hmm.
So one simple test would be is to do a 96 correlation, right, from storosporin after calibration with DMSO on each plate, right? So you do an all versus all scatter plot, right? And you'll have lots of scatter plots, but you'll see they should all basically be a good correlation. And you do it before calibration and after calibration. So those which are just basically the raw intensities versus that when you calibrate it to the DMSO.
To the DMSO, yeah.
You have to be careful though not to just divide by the DMSO. You have to divide by the DMSO and then multiply by either like the maximum or the medium across all samples, right? So by a constant value.
Yeah.
So you spread the intensities out.
That'd be cool.
Anyway, I think that would be a great figure, and it basically shows it would be the same sort of thing, and showing that basically it doesn't really matter whether or not things were done in plates adjacent to one another versus through 6 years apart. They basically give the same results.
No, I think that would be important to see.
Yeah. Anyway, I think that's basically that. I mean, is there anything else that we'd want to cover as far as so I think we can actually add another topic of something to do, and that is data harmonization between groups. So Jenny, given the fact that you're working on CSF, we'd like to send you some of our reference CSF, and maybe we can then see whether or not we can compare the assays between cerebral spinal fluid that's generated there versus.
Yeah, we're running CSF is being run on the thermo instrument on the Explorer. So I think we have one we have plasma being run twice, one on 6600 and one on the Explorers, and then we're running CSF on the Explorers. Yeah, I'd love that. That would be super fun. It would be very good.
In our U19, Parkinson's disease is the control, right? So, because we want Lewy body disease as the control for the Alzheimer's disease. So it would be a good way in which we could potentially bridge between data sets.
I would like that very much.
Yeah.
Great idea.
So we should. So have you? So we should get that to early.
Yeah,
on.
we're starting to run. Probably, my guess, we were down because of COVID-19 in our lab, so we'll be starting back up. Probably give us 2 weeks before we'll start having stuff in. But if you could send it in the next week or two, we'll start getting it. We'll get the digestion done because we should do batch digestion as well on it. Yeah, that would be really.
So I can also, we do have a CSF reference and we have a CSF QC, and our QC is very different. It's basically middle-aged, young, healthy people, right? And it's very different from the other one. So we can send you both.
Yeah, that would be great. We can send up with the ones that we have for AMPD are, we have controls for the different cohorts. So also control and negative, control and disease. Okay. So it would be interesting to cross over actually. So why don't we talk about that offline? I think it would be really, really cool.
I just want to extend, so I think that's obviously, but we can do the same thing with Malvina's data set, right? We're basically going to we're using the samples that she had sent us to demonstrate we can calibrate to the Broad data. So yeah, so I think those are, so we have the same, obviously the same DMSO that you've run on one of your DMSOs, and we can use that as a calibrant. And so I do think there is value to demonstrate this ability to calibrate between labs and data sets and time. Right?
I think it's part of showing maturity of a platform more so than some of the platforms that are being used now, right? So I think it's moving it, because I think both of you and I are so influenced by clinical chemists that I think we have to push that, right? If we ever hope to get stuff into the clinic ultimately.
Yeah, so I think those are things that can be done in the upcoming, and anyway, does anybody else have any? I don't want to be.
Maybe if Ajay is here, maybe he can just say a few words. I don't know if he's still.
He left, I think.
He left.
He
Okay.
left.
I wasn't.
Yeah.
Then I think I will say, on behalf of all of us, thanks to Andrea and Mike for organizing this. I think it's been really good. Thank you.
Okay. All right. Thanks. It's been, yeah, I guess I'm looking forward to seeing the other talks from the other groups.
Yeah, me too. I think it'd be really, really good. It's time.
Awesome.
The bell rang.
2 minutes, it's 2 minutes.
Thanks, everyone.
Okay,
So it's nice seeing everyone.
Yeah.
Have a good evening.
All right.
Thanks, Andrea.
Thank you. Thanks,
Take
Jennifer.
care.
Bye, Andrea.
Bye.